So my name is Luigi Jovane. I'm professor at the Institute of Oceanography of University of São Paulo. And I'm going to give you a talk on magnetotactic bacteria and how they are related to the environment and which are the clues to understand past environments, today's environments, and which are the information about the carbon cycle that can give to us for today, future, and the past. So I thank Frank for the invitation and Aldo for the invitation to participate to these seminars. So what are magnetotactic bacteria? Some bacteria, and this is in blue, there is an image of them, which can produce internally very small crystals of magnetite. Sometimes are also graphite, and we are discovering that actually there are sometimes also other magnetic minerals. These crystals are not always in the same shape. So here you can see, for example, some different shapes of these crystals that are produced internally from the cells. We think that the main reason of them to be made inside the cell is to move through the geomagnetic field, Earth geomagnetic field, through the sediments or through the water to get the ideal conditions. So this is the main reason, and this is called magnetotaxis. So what happened is that these cells then died. So the bacteria died, and the magnetosomes, which are the crystals, then that get dispersed in the sediments. And here are some images of these magnetosomes, which are the crystals that are dispersed in the sediments after that the bacteria died. And you can see that they are very different in shape, in dimension, and in the morphology. There are some which are actually very complicated. They looks like a star. Some are really long, elongated, and we have some clues that maybe are not actually producing internally from the bacteria. But what is really interesting today is that there are some events, and here is the first recognition of this magnetotactic bacteria as magnetosomes 55 million years ago during the PETM, which was a very warm and abrupt event that occurred globally and have been discovered firstly in 2008, the occurrence of magnetosomes, of different kind of magnetosomes along the warmest period of this event. And there was also some relation between the length of the crystals and the dimension of the crystal. So they were able to also discriminate through the magnetic properties some features of the morphology of the crystals, and was a very nice attempt of relating magnetosomes with the environment. Later on, these are some other papers that recognize in another event, it was also a very hot event, the Miko event 40 million years ago. We recognize a lot of magnetosomes in the sediments all over the world. This is in Monte Cagnaro in Italy, and we recognize these crystals. And actually, what we recognize actually that is there were two main populations of magnetosomes. So through lab measurements, magnetic lab measurements, we were able to discern two main populations of crystals, which probably were related to two different populations of magnetotactic bacteria. And actually, we recognize that was the occurrence, through the occurrence of the MECO, that firstly was divided between the pre-MECO, the MECO clue, and the post-MECO. We recognize that these magnetosomes were different along this event. And here are some forks. Forks are first-order reversal curves, which are made by a paleomagnetist. And when they show this feature, this elongated structure at the middle of it is unique and is only related to magnetosomes. So the magnetosomes lost in the sediments give also very small quantities, can give this signature. And so this signature is really the proof that magnetosomes, magnetotactic bacteria was living at that time in the sediments, because we find magnetosomes. This is another core, always from the MECO event, which shows again as a big peak of magnetic properties which are related to magnetosomes, again in two main populations, so different magnetizations and magnetic parameters along the event. So this was another proof that actually globally, during the warm events, we had a diffusion of magnetosomes all over the oceans. And this the fork that I was telling you, so this is the characteristic feature of a fork. And this event was also studied in Turkey, and we find also here with very high resolution, because this is a section of over 350 meters, and the MECO is expressed in 60 meters, while everywhere in the world is around 1 meter or a bit more. So this is very expanded section, and here we were able to characterize the MECO through stable isotopes recognized in different kind of organism living originally at different depths. So this is the trace of the story of them. And so we can see that actually after the MECO, there was a strong stratification of the water masses. And this is also confirmed by the study with the clay minerals, which also show this huge stratification that occurred just after the MECO. So the MECO event, this warm event, was a revolution for the ocean stratification. Exactly on the MECO, there is a spike in the magnetic properties, which is related to magnetotactic bacteria. And what we understood so far is that actually what is happening is that iron and manganese that are abundant in the seawater, what happened during the MECO is that this event is characterized by low pH. So the acidification of the water make increasing the manganese in the water column, and increase, and do not deposit because of the acidic condition. And once the warm event finished, and so we reestablish again the normal pH condition of the ocean, we get the huge deposition of manganese. And this is the main story that we know about with high resolution of the MECO. And this is very important because give us information of how nutrients are important for the presence of magnetotactic bacteria. So what we can say is that magnetotactic bacteria are not just organisms that lives in the sediments due to some nutrients and some conditions that make them being abundant. But actually, magnetotactic bacteria can be living in the sediments through time in different position of the metabolic processes along the sediments in relation to different metabolic processes. So there are probably population of magnetotactic bacteria that like condition with oxygen, others that do not like condition with oxygen, like pH, low pH, other ones that do not like low pH. And in the same way, manganese and methane can be very important for some kind of population of magnetotactic bacteria. So there are many magnetotactic bacteria. That's the main story. And they can be seen now as a real primary productivity inside the sediments. Okay, so they don't just use nutrients and their second primary productivity, but their primary productivity related to some nutrients and metabolic process occurring in the sediment. So there are mainly several points that make the presence of abundance of magnetotactic bacteria. So there are iron, grain size, poor oxygen in the poor water, minerals and sediments. So we try to understand which was this of these topics, which was the most important for the presence of magnetotactic bacteria. And what we did was we did a research in Saco de Mamanguá in Brazil, a very nice place, of course, but it was also very particular because it's very, it's area, so it's a very quiet place with a very slow sedimentary rate and not a lot of energy. So it's mimic the condition of a warm period because there is a lot of clays, a lot of nutrients, height hydrological cycle, height carbon cycle. So this place is very abundant of magnetotactic bacteria. And this is the map of it. It's between the state of São Paulo and Rio. And it's also unique because I don't know if you can see here through this core, there are a lot of small bubbles on the top of the sediments. This is methane. So magnetotactic bacteria somehow, or they like methane or they produce methane. They have some metabolic process also related to methane, and this place is bubbling methane in very in some period of the day. So and we did several cruises of studying in this area. It's completely preserved, so no anthropogenic impact related to other things. And we were able to collect a lot of samples with different magnetotactic bacteria. So it was really interesting, and some of them are new and will be showed in the future. What we studied is actually magnetic properties along the core. So here are these magnetic properties, which shows that there is a huge change only in the first 40 centimeters. And the first 40 centimeters is also the interval where we have highest oxygen in the sediment. And so we started thinking that it was a relationship between oxygen and presence of magnetotactic bacteria. And so we studied with a very high resolution the magnetic properties and poor water and everything we were able to study along this interval. And actually what we recognize is that going deeper, the Curie point, so you know that the magnetite lose its magnetic properties, became paramagnetic at 580. But actually, this temperature decrease when we get deeper. And this is related to the presence of magnemite. So there is oxidation of the crystals of magnetite due to the oxygen that start to be depleted going deeper. And this is very clear with synchrotron experiment, which we point exactly on the crystal, and we saw that there was this is called Xanes. It shows very clearly that magmite start to build and change the crystal of magnetite. And also through forks that definitely show magnetosomes, presence of magnetosomes, but they are different. You see, this is not exactly the same fork. It's shifted from here is 20 millitesla, you get to a spike around 35 millitesla, which so it changed a lot, and this is related to this change of the crystal of magnetite, which get oxidized, and at the end, below 80 centimeters, there is nothing more. The all magnetite is completely destroyed by other metabolic processes. So, so the fact that we get magnetosomes during this warm period is really particular. It's very important to be understood. And we studied in several cores, and all the cores shows the same features. So this is a core, and we get up to 34 centimeter presence of magnetosomes. In this one, a bit shallower, but then we had some more here around 50, and in this one around 30. So all cores shows the same features. And actually, also giving more characterization of this fork data. So this is the fork of biogenic magnetite, which is this light gray part of the cores, completely disappear after 20, 40 centimeters. Below of this, we only have detrital minerals, magnetic minerals, which are actually characterized by these other end members in the forks. And these are constant, so they remain constant. So magnetite, also if it's produced by magnetotactic bacteria, it can be dissolved very easily just after a few centimeters in relation to the presence of oxygen. So just to start closing the story, we can see that there is no changes in the mineral composition, no changes in the grain size, and no change in the sediment characterization or petrophysics. And so we can exclude all of these topic features. But then when we start studying the pure water chemistry, oxygen and oxygen reduction potential and dissolved iron, we actually see that the main features are actually this oxygen reduction potential that it change in the upper part, exactly where magnetotactic bacteria are present. So what we can see is actually that, and this is also very important, that the bacteria, they produce magnetite in a field that actually is stable for goethite. So the cell is able to preserve the crystal until the bacteria is alive. When the bacteria died, then it start to get dissolved, first as a magmite, and then it completely disappeared when it entered in this other field that is related to the oxygen reduction transition. And so we can say that iron actually is not an important factor, and as you can see here, is constant along the core. But what actually is really important is the oxygen. And what we also studied at high resolution is the IRM acquisition, which, as you can see here, the components recognized, so this population that I started talking to you at the beginning, are always present, different population. One is called soft biogenic, other one is called hard biogenic, and they are often present, but it's clear that they change along the depth. And there is a particular depth, and which is related to the oxygen, the presence of the oxygen, which is the oxidation reduction transition, below which the two main population of the hard and the soft biogenic magnetite start disappearing, and one of them completely disappeared. And what we get then is only the detrital magnetite that was originally already there, but then is the primary magnetic component. So in synthesis, iron is not a factor for the presence of magnetotactic bacteria. Grain size, not. Minerals, too, is not a factor. Sediments or the parameters of the sediments, petrophysical characteristics are not a factor, but mainly is the oxygen. So what make actually, what is important related to the presence of magnetotactic bacteria? So one of the factors that we find very important is to translate it in numbers. So how much magnetotactic bacteria, how much magnetization? So we were able now to start understanding with this bacteria that has been collected from the field and cultivated in lab. And here are some images of them. Here are the bacteria with the magnetosomes inside, and here are the magnetosomes lost in the sediment. And so we were able to count the number of cells. And this was really revolutionary because we were able to define, and here is the main plot of this recent study, which is the edge of sensitivity of number of cells that we can recognize with magnetic properties. And here is two different species of magnetotactic bacteria cultivated, which both increase their concentration and is visible in the magnetic properties only at the highest amount. So this lowest amount of number of cells, also if it's 2 is 2000, 4 is 4000, it's already a big number. But our equipments are not sensible enough to detect the presence of magnetosomes, and only when we get to 7,000 or 9,000 we are able to detect them. These are the forks, and so these are two methods, and both methods so to show that actually these are not artifacts, shows the same feature. So here the peak is around 25 millitesla for this species of bacteria. For these other species, around 40, 50. So much stronger and completely different also in dispersion and in coercitivity. So these two species are completely different in magnetic response. So this is another example how we can characterize magnetotactic bacteria also through studying magnetic properties. And here are the numbers related to actually this count. And so we were able here to give a value, a real value of how much carbon is preserved in relation to the number of cells that were counted and the magnetization that is measured through magnetic properties. And so we were able to make a direct relation between the amount of cells of the magnetotactic bacteria and magnetization. which is an incredible resolution, because if we apply this method for the past, we are able to say how in this past warm event, how much bacteria, magnetotactic bacteria, was present in the sediment at that time. So this is a really revolutionary study, which provide these new numbers that can be used for modeling of paleoclimate studies, and can also give information about the application of magnetotactic bacteria to maybe sequestrate, sink carbon in the ocean, in the sediments. So in conclusion, oxygen is the concentration is the main controller of dissolution of biogenic magnetite. So it's the main parameter for the presence of magnetosomes, and which are actually much more complicated than what we know. So we already know that there are different populations. Some are soft biogenic magnetite, other are hard biogenic magnetite. And there are different methods. I show you forks. There is IRM decomposition that also can be used to show the presence of magnetosomes and population of magnetosomes. But more importantly, now we are also able to give the threshold of sensitivity of magnetosomes, presence of magnetosomes in the sediments through a study of polymagnetic studies of in labs, and so we can be able to reconstruct past abundance of magnetotactic bacteria, and of course give information for the global carbon cycle, which is a very important topic for today's changes. So we hope that this study can be really used for improve these researches. So I wanted to thank, of course, the group for you to be here, and my lab, the core, LECOM, which is at USP, which is the laboratory of ecological microbiology, and the laboratory of microbiology in Rio de Janeiro, which do the image of the dam and do also the cultivation of the magnetotactic bacteria. And of course, financial support from FAPESP, the Foundation of São Paulo, and CAPES, the Foundation of PhD students in Brazil. Thank you very much.